I'm happy to see you all here. Today will be Mort. He is currently visiting us from India and staying here for months in a group. He's going to present some of his previous work. Please go ahead.
Yeah, thanks.
We're happy to have you here.
Hello everyone, and thank you for coming. I'm from Indian Institute of Science. I'll be presenting two of my works. One was closely done with Swedish group and one was independent. We generally get scientific data represented as a scalar function over a given geometric or spatial domain. There are various techniques to visualize it in topology, and some of the structures that are majorly used are Reed space, fiber surface, Jacobi set, continuous scatter plot, feature level sets. The paper was from Ingrid's group. I majorly have worked on Reed space, fiber surface, Jacobi set, and continuous scatter plots. The first one is just a univariate precursor work when I started my PhD. Second was a short paper, and its extension we have submitted in TVCG. First of all, what do I mean by univariate? If I'm given a spatial domain, say here I have a two-dimensional domain, then if I define a scalar function over each point, say this is value for this scalar function at this point, this is at this point, then I call it a univariate scalar field. If I want to find out all the points which has a specific value, then all those points we call as level set or isosurface or isocontour. In that left image, those black curves, they are the level sets. A level set can have multiple components. A single component of that level set we call as contour. Critical points. The points where level sets change their topology. By topology, I mean they either emerge at that point, or they vanish at that point, or they merge or split at that point. E and O are two maxima. J is a saddle point. Multiple contours are either joining or splitting at J. Next is contour trees. Contour tree basically captures the evolution of these level sets. Contours emerging from O and E are joining at J. That thing we can observe that these two arcs are joining at this saddle. Next we move to multivariate terminology. Instead of one field, we deal with more than one field, and most of my work has been with two fields so far. Here in figure A, I have one field which is ranging along z-axis. Minimum is at the back and maximum is at the front. Second field is a distance field which just captures the distance from this origin of spatial domain 0, 0, 0. In C, this is called continuous scatter plot. It is nothing but a continuous version of discrete scatter plot. It is basically a two-dimensional histogram which just says that at this particular point in this histogram, the density, which is a measure of occurrence of that bivariate value, that is captured by this color map. Because near the origin I am having darker color, that just says that lot of points in this bivariate field are there which have 0, 0 value. In D, we have two different isosurfaces. Yellow one is the isosurface for a particular distance value from B. Then the pink or magenta surface is isosurface corresponding to the second intersection of these two isosurfaces. Black curve, that actually represents all the points where one field has a particular value 0.5 and other field has a particular value minus 0.1. In continuous scatter plot, that is represented by this black sphere. This black line or black curve is called fiber. Fiber is exact alternate of isosurface or isocontour. So it just says, give me all the points where you have this bivariate combination. That is minus 0.1 for one field and 0.5 for other field. If I vary this black sphere continuously across this blue curve or these two lines, then what that will be representing inside domain. Instead of just a fiber, all these points which are lying on these two lines will be representing a surface, and that surface we call fiber surface because it is a collection of multiple fibers. That surface is represented in figure E. It is passing through that black fiber because it is supposed to pass. Next is Jacobi set or Jacobi edges are exact alternate of critical points. Critical points, we said that the points where level set topology is changing. In bivariate setting, the edges where fiber topology is changing. Fibers are either merging or vanishing or splitting or merging. If I take one restriction of one scalar field, I fixed one scalar field which was along the z-axis, and I have computed contour tree on just this restriction, contour tree for the other field. If I stack all such restrictions and I compute contour trees for all those restrictions, I get such a network of edges, and these edges are called Jacobi edges. They can be imagined as concatenation of all these critical points computed over all the restrictions. Then Reeves space, if we stack all the contour trees, we get a Reeves space, and this Reeves space divides our domain, our bivariate field, into segments of same topology. What do I mean by that? Same fiber topology. I see 3 different sheets in this Reeves space. Each sheet belongs to a segment in domain. Inside each segment, I'll have similar fiber topology. This blue, red, and white. What do I mean by similar fiber topology? If I compute a fiber, this particular one, the same minus 0.1 and 0.5, when I keep on increasing, say, one particular value, at some point, I'll cross this green surface. This green surface corresponds to this green Jacobi edge. We call it Jacobi fiber surface. As soon as you cross this green surface, the fiber topology will change. That is, instead of one connected component, I'll have 2 fibers now. This is how this Reeves space is a useful thing. It lets us know which are the different segments in domain which are behaving as same. This is projected version of these segments. I just want to see how these segments look like in the projected range space. By projected range space, I mean one axis is one field and other axis is other field. So it is just visualizing where my Jacobi edges or where my segments are going inside range space. How this bivariate analysis gives me an upper hand over the univariate analysis. So this is ethane diol molecule, and I want to identify different bonds in this molecule and different atoms in this molecule. I have 2 different fields here, electron density and reduced gradient magnitude. Using univariate analysis, I can select a particular threshold on, say, electron density that gives me all the 4 atoms, and on the gradient magnitude I can select a particular ISO value that gives me all the bonds. But using univariate analysis, I am not able to identify which are oxygen atoms and which are carbon atoms. If I plot this continuous scatter plot, I see these 2 distinct notches and 2 these parabolic structures on top. So these are 4 distinct characteristics in this continuous scatter plot. The bottom 2 belongs to different bonds, and those 2 parabolic structures, one belongs to the 2 oxygen atoms and one belongs to the 2 carbon atoms. So we are able to separate out 2 sets of atoms if we move to bivariate analysis. Studying electronic transition. In this application, we had 2 fields. One was whole NTU and second was particle NTU. So a particular state is given to me for that molecule. Electronic transition happened. Whole NTU tells me how much charge at each point in that molecule was lost. Particle NTU tells me how much charge at each point was gained. I want to analyze how strong acceptor a subgroup is. Finding different regions inside that particular subgroup. Maybe one subgroup is behaving as a donor and acceptor both. So I want to identify which region is actually donor, which region is actually acceptor. So these type of problems we were trying to solve. In order to study this thing, we gave 2 different operators. This vertical line separates the 2 operators. We call both of these CSP operators, continuous scatter plot operators. In first one, we just consider continuous scatter plot of that particular molecule, of that particular state. X-axis is telling me amount of charge lost and y-axis is telling me amount of charge gained. We use the continuous scatter plot and we try to divide or segment that scatter plot into donor and acceptor parts. We call them CSP lens operators. This operator is donor operator. It will just extract regions where charge lost is more than charge gained. We get some scatter plot, and in the same way we get scatter plot for acceptor. If we want to see where exactly this donor and acceptor is lying, we simply use fiber surfaces. We want to see, say, a particular donor strength. Where is this particular donor strength? This is a constant donor strength curve is lying. I just select a control polygon here. In this particular conformation of molecule, this is thiophene subgroup and this is quinoxidine subgroup. This particular subgroup is donating, and this particular one, because it has acceptor CSP, it is behaving as an acceptor. Next was CSP peel operator. If I don't want to know the global behavior, or I don't want to extract globally in the molecule, I want to extract a subgroup level behavior. Just tell me how this subgroup is behaving. For that, what we do, we simply segment the subgroups. We segmented this subgroup in one spatial domain or one bivariate field, and we segmented this one in another. And we plotted CSP for individual subgroups. We found out that if we plot CSP of this is aligned with x-axis, that is charge lost at each point is more than charge gained. This subgroup is behaving as donor and quinoxaline is behaving as acceptor. So these were the two main operators that we gave. Both of these operators can be used independently and together. On x-axis, I have all the length operators and I'm interested mainly in say donor and acceptor. On y-axis, I have CSP peel operator. Now together these operators, what information they are giving me is, say, I just want to analyze quinoxaline. Quinoxaline's CSP is this. Now this CSP is not telling me whether it's a donor, acceptor. If I want to identify donor and acceptor inside this particular subgroup, then I apply length operator on this CSP itself. And I found out that, okay, this is the donor CSP and this is acceptor CSP. If you want to see where exactly inside space these regions are, I can select control polygons and those control polygons will give me the spatial mapping. Using these operators, first we studied thiophene quinoxaline's behavior with different geometries. By different geometries, I mean these are two subgroups. First is thiophene and second is quinoxaline. In 0 degree configuration, the dihedral angle between the planes which are containing these subgroups is 0. Then we keep on changing the angle 60, 90, 120, 180. What we found out was that 0 degree conformation and 180 degree had almost similar CSPs, which was kind of expected because they are almost similar except for this yellow atom. And as we increase the angle, the amount of charge which is being donated from thiophene to quinoxaline, it keeps on decreasing. Here it was high, at 60 it decreased, at 90 it was minimum, then at 120 again it started increasing, at 180 it became similar to 0 degree. That is one relaxed geometry in the end. Here we don't constrain any specific angle on the molecule, we just observe how does it behave in a relaxed state. These quantities, they give me the measure of how much charge is donated and how much charge is gained. Here we have two copper complexes, Cu-PHE pheon and Cu-PHE Zen. For both of these we have two states. We wanted to identify in which state charge transfer is happening, that is charge is being transferred from one subgroup to another, and in which one local excitation is happening, that is charge is being transferred from that subgroup to itself. Again, by seeing the visual representations, we could identify that copper is behaving as strong donor here, PHE is behaving as strong acceptor, and the third ligand, which is pheon here, is kind of inactive, it is not much participating in this. For the state 10, PHE CSP looks exactly similar as complete molecule CSP and other two subgroups are almost inactive, which says that PHE is the one which is participating in this excitation. This is the recent work, although it started before my previous work, but it was submitted now. What we aimed initially was to have a better method for computing fiber surfaces. This is the existing method. What existing method does is, it traverses complete domain, complete spatial domain, and checks each tetrahedron in that domain, whether the required fiber surface that I'm searching is crossing from that tetrahedron or not. So it is going through all the tetrahedron. We just wanted to avoid this and somehow reach to the exact location where fiber surface is lying. If I am given a contour tree here and I want to identify an isosurface corresponding to this particular iso value, this line is intersecting these two arcs, which means that if I start traversing my spatial domain from these points C4, C3 and C5, the desired isosurface will be lying somewhere nearby. This contour tree representation helps me to reach to the isosurface faster. We wanted to use the same idea. The challenge was that exact alternative of contour tree will be Reeves space in bivariate, which has its own computational challenges. And we relaxed that requirement that can we use instead of Reeves space just the Jacobi edges. In univariate setting, can we just use the critical points instead of complete tree? This is my same projected space which shows me all the Jacobi edges. I just know that these are the edges. I don't know any connectedness between them, how those edges are creating a surface inside Reeves space. First challenge was to identify the required Jacobi edges, which will help me to reach to the fiber surface faster. This UV is the control polygon. In order to find out the interesting Jacobi edges, I just extended a line through this control polygon edge and found out all the Jacobi edges which are being intersected by this particular line. This line will actually give me more number of Jacobi edges than exactly required, but it will definitely give me the Jacobi edges which are exactly required with some extra information. So first step in our method is just this: find out all the necessary Jacobi edges. Next is, from each Jacobi edge inside domain, I do a directed search. Directed search means I start traversing the neighboring tetrahedra and try to reach to the control polygon edge, or this U and V bivariate values inside domain from each of the edge. I just move closer to UV. As soon as I encounter U or V, I get to know, okay, this is the tetrahedron I require. This is my seed tetrahedron, and I'll be traversing my fiber surface, or I'll be extracting my fiber surface from this seed. So using these directed searches, we identify seeds. Next steps are very simple. We have the seeds, we just explore the neighbors, and we extract all the tetrahedra which are containing that fiber surface. Qualitatively, it works very well. Results are exactly the same. We compare visually, and we also had some numbers to compare, that is how many tetrahedra our fiber surface is passing through and how many are exactly required. Those matched. Performance wise, there were some issues. TTK implementation, they have an advantage of octree based method. So they can divide the space, they can use octree. Definitely that works faster. Without octree version, in most cases, our method works better. Our method works bad where number of Jacobi edges increase, because the method is dependent on these finding these Jacobi intersections. So if number of Jacobi edges are almost same as domain size, we are spending that much time in identifying those intersections. In terms of application, we are able to give something new, that we are able to compute fiber surface components flexibly. Previously, for isosurface, there exists such a method. Say in this contour tree, it is intersecting two arcs. One arc will give me one component, isosurface component. Other will give me another isosurface component. For fiber surfaces, there was no such method which gives me individual component of that fiber surface. Because this method is based on Jacobi edges, it was able to give this thing to me. Let's consider this example. Here, this red one is my Jacobi edge. The line is intersecting all those edges which are highlighted in red. When I map those intersected edges inside domain, in domain they are lying here, which is exactly the same location where oxygen atoms were, and this is where carbon atoms were. So if I select a particular Jacobi edge in this cluster, I can extract one oxygen atom instead of two. Same thing for other components. If I select, say, one Jacobi edge from this cluster, I extract one carbon atom. From here, I can extract one center component of that bond. This was one interesting thing which came out of this technique.
Tooth, is it? Is that bivariate?
Yes. Where did you see that?
The previous slide.
Oh, okay.
I didn't know it was.
There are no two fields. We can have some derived fields. So in tooth, they use gradient, the second field.
Oh, okay.
Generally, to extract the surfaces, the gradient works very well.
The second question then. The worst case for this speeding improvement would be the field is uniform random.
That is the major point. Our algorithm will perform bad when there is a lot of noise and a lot of disturbances, and the Jacobi edges itself are maybe exceeding the number of tetrahedra in that domain, or maybe near to that number. If it is very structured, then our algorithm performs very well. Atom?
AtomDL2, purple blobs there.
Oh, this?
AtomDL.
Oh, this one?
Yes.
This is also ethane diol dataset, but the control polygon selection is very weird. Here, the control polygons belong to some specific features which are interesting features. This one I just selected to highlight that our algorithm can actually perform good. Here, if you see 898, if there is some specific case, octree may perform bad.
From your first paper is that you could use your kind of method and then you can kind of showcase like where on atoms. So is this like isosurfaces or?
I'll go a little bit in detail here. You can consider it as an isosurface if your field is donor strength. Then it's isosurface of donor strength. If my field is bivariate, that is whole NTO and particle NTO, for those two fields, it's a fiber surface. So I'm selecting a control polygon here, and corresponding to those two fields, I'm extracting a fiber surface. But eventually, it's an isosurface of donor strength because both the fields are related.
You had this nice picture in correspondence with the contour tree, where you draw this line and say, okay, here, exactly, I have two components for my isosurface, and I probably want to select this and that.
Yep.
And you draw this analogy. Do you have an idea about how many components your fiber surface would have without computing the entire reef graph?
If I have the reef space, if I have this thing, if my control polygon is selected in this region, and I know that this is one particular sheet that belongs to the control polygon. If it is selected in this particular region, okay, one component here, one component here. But if I don't have this information, then about exact components, we don't have any idea, and we are completely relying on control polygon we have selected, and corresponding to that control polygon, we can say, okay, maybe 4 components, because all the Jacobi edges are forming this nice cluster.
The number of Jacobi edges, would this be an upper limit?
It may happen that you select a control polygon and that is not intersecting with any Jacobi edge. It is just part of some small tetrahedron.
Then you have no guarantees with your method that you find everything.
Guarantee is there. It is not that Jacobi edge is supposed to intersect a control polygon edge. The line is supposed to intersect. So for sure, even if there is a small component, we will be able to identify it. But how many number of components will be there?
So you can have a lot of lines.
Yeah.
I'm just curious, like how this search is done. So you say you go towards this control polygon.
Just each tetrahedron, just imagine that is mapped somewhere in this range space. So I see where each edge of that tetrahedron is intersecting with this line, and which is the closest edge.
Wow, you
and I
actually
keep on,
implemented.
yes, I keep on moving forward from that.
So, seems like there are no questions anymore, and the hour is over also. Thanks again for your talk.
Yeah. Thank you.